Dr. Uh, oh, just a sec. So Dr. Bhattacharya is our distinguished speaker today. He is a senior principal scientist and the head of molecular genetics division in the CSIR Indian Institute of Chemical Biology, Kolkata, and is also a professor of biochemistry and molecular biology in the Academy of Scientific and Innovative Research, New Delhi. Dr. Bhattacharya got his PhD in life sciences from Jadavpur University, Kolkata, and did a very successful stint as a postdoctoral fellow in Friedrich Miescher Institute in Basel, Switzerland. And thereafter, he joined CSIR IICB in 2008. And since then, he is continuing his remarkable research on mechanism and consequences of miRNA activity and modulations in mammalian cells. So till date, he has a total of 43 publications in many prestigious journals like EMBO, Molecular Medicine, Nature Communications, EMBO Reports, Molecular Biology of Cell, Journal of Cell Science, etc., with over 12,000 plus citations. He has received many national level awards, including the most prestigious Shanti Swarup Bhatnagar Award and the National Biosciences Award, and also Swarna Jyoti Fellowship from Government of India. So today, Dr. Bhattacharya is going to talk about mechanism of export, trafficking, and uptake of extracellular vesicle-associated miRNAs in mammalian cells and tissues. We extend a very warm welcome to our distinguished speaker today and request him to deliver his talk. So Dr. Bhattacharya, please go ahead. If you need us to adjust anything, let us know.
I think everything is fine right now. So thank you, Susmita and Bhavatos, also for inviting me to this wonderful seminar that I'm going to talk about in this platform. THSTI is a very good place to do cutting edge science for the translation research, as I know few people there already, like Bhavatos and others. It would be nice if we could go there for a offline interactions and meeting, but as the situations stand right now, it's sometimes difficult to plan that thing. So with that limitations, I start my seminar, but I think I will be able to communicate the science we have done over the last a decade, I would say, in IICB Kolkata after getting my independent position there. And as Susmita mentioning, most of the achievements we have done in this microRNA field are primarily done over the last 12 years in the Research Institute, Indian Institute of Chemical Biology, where my lab is now. We call proudly as RNA Biology Research Laboratory. So with this introduction, I like to switch to the topic. So as already mentioned, that is a mechanism of export, trafficking, and uptake of extracellular vesicles associated microRNAs in mammalian cell and tissue. So before I proceed into the in-depth and talk about the extracellular vesicles and its as a role as a communicator of gene signal across the cells and tissues, I like to introduce you to the microRNA world. So microRNA are 22 nucleotide long regulatory RNA, as we all know, and they are processed inside the nucleus from its precursor like pri-microRNA, and then to the pre-microRNA exported out into the cytoplasm, which is processed further to the mature form by two protein dicer and TRBP. And these mature microRNA, when becoming double stranded one of the strands encoding the mature microRNA get incorporated into the argonaute proteins, and this argonaute microRNA complex, this find the target genes where the microRNA can find the complementarities and repress the target genes by translation repression and also induce degradations of the message. So that is in nutshell is the way the microRNA functions. There are more than thousands microRNA are predicted to operate in humans, and as you know, one microRNA can regulate several genes. In that way, almost 50% of human genes or even more are predicted to be under microRNA regulations. And by the way, that microRNA regulate developmental process, differentiation, apoptosis, and so many processes that are prevalent in the human cells or in higher animals. Expression is dysregulated in human diseases. And of course, the developmental and tissue-specific expression pattern of different microRNAs have been observed. Like there are microRNAs that are expressed in brain, there are microRNAs that are expressed in liver exclusively or in the muscle itself. So, what are the factors that control this kind of microRNA that are known to be the post-transcriptional gene regulators present in the higher eukaryotes? How they controlled by the other factors? What are the factors that control the microRNA activities in the mammalian cells? So, in this context, one of the interesting aspects of microRNA regulations comes into alive when people have discovered that extracellular vesicles is a means of carrying microRNA between cells and between tissues. So I just like to introduce the extracellular vesicles a little bit. So these are formed through the endocytotic process when this endosome mature into the multivesicular bodies. These multivesicular bodies fuses with the plasma membrane and release their content. And it has been found that these small tiny vesicles that are part of these multivesicular bodies, when they are released in the extracellular space, uh, called primarily as exosomes, which are around 100 nanometer of size. They can carry different things, including some RNA like microRNA, but they also carry proteins, different kinds of lipids, and as well as mRNA. So people consider them are good biomarkers, vesicles, because they are representing the cell situation or cell status, because they carry some of the material inside the cell to the outside milieu and be part of uh, the serum and other biofluids. So they can be used as good biomarkers, people consider them. But on the other hand, they are considered as good cell-cell communicators, because they are communicating these extracellular vesicles released to the other cells. And by the way, they communicate between the cells, and they can even traffic across the tissues to reach the other tissues where the exosomal content get delivered, and that way they can communicate between the tissues as well. There are different kind of extracellular vesicles. Exosome is one of them, but there are microvesicles that can also carry some content. But sometimes microRNA itself get complex with albumin proteins or other kinds of molecules like HDL particles. They have people identified these are also can carry microRNA outside the cells, and they get trafficked in between. So role of extracellular vesicular microRNA in disease is a very important thing to look at, because you can imagine when the microRNA can get exported out of the cells, they can be a major player in controlling the microRNA activity from the donor cells, because they are getting packed and exported out. On the other hand, they are taken by different cells also, in that way that are representing a way of communicating the epigenetic content between the cells, and microRNA level can be also altered in the target cell because they are getting delivered there. So there are mechanisms of microRNA packaging into the EVs. There must have specific mechanism. There must have factors that affect the microRNA loading into EVs, and there must be factor responsible for the intercellular microRNA transport. And of course, we have interest to understand the mechanism of this kind of microRNA that are released by the exosomes or extracellular vesicles into the target cells, how they get loaded into the recipient cell and become functional there. So these are the questions that I'm going to try to answer some of them in the next half an hour or so. So you may see we start with the questions how the target RNA presence may affect the microRNA turnover. So as we know, the mRNA is getting targeted by microRNA. As when they are targeted by microRNA, they are getting through some kind of translation repression or degradations. But in the reverse, we ask the questions how the microRNA that are getting involved in this repression process are get altered by themselves, whether there is a specificity of any kind of usage specific turnover of microRNA happens. So how is the usage of microRNA is related to its turnover, whether the pool of microRNA that are used for the repressions are more labile or more unstable than the reserve microRNA pool that are not used for the repression to occur. So to address the questions, we did some experiments in which we use renal reporters, luciferase reporters, and these renal luciferase reporters usually have uh, also can have the UTRs with the microRNA binding site. In this case, a LET7A microRNA binding sites. The binding site could be of different types. This is the seed region where the microRNA and the mRNA target mRNA binds, and this is the anchoring regions which can have different complementarities, and in that way we can see that depending on the complementary and the size of the bulges or the imperfect complementarities, we can have two kinds of things where have A and B, we named it. But in both cases, we find that there is a repression or lowering of microRNA late 7 happens when you have this reporter RNA in excess in the cells. So that means when these reporters are getting repressed by microRNA, they are also inducing some kind of turnover or degradation of the respective microRNA late 7, whereas the other microRNA mid-16 didn't get affected. And it's not happening in only one cell type in HuR, but it also happened in another cell type MD-MB231, and it's not a cell type specific event, but their turnover is controlled. We did further experiment to use some non-translatable analog of the mRNA, reporter mRNA, which cannot get translated or have a slow translation, to show that the active translation of the target mRNA is required for the microRNA lowering. This lowering effect we observed here is actually contributed by the translatability of the reporter mRNA, and that's with the repression by the microRNA itself. So microRNA lowering ability depends on the microRNA mRNA binding site, as we already mentioned, that A and B, there are difference also in the lowering type. For example, the A type show less lowering, whereas the B type show higher lowering. So this is also contributed by some kind of stability or complementarity between the microRNA target sites and the reporter mRNA. In the next experiment, we have to find that target-driven export of the used microRNA actually happens. That means the microRNA that are repressed by the target that used for the repression are more preferentially get exported out. So we did the same experiment with the extracellular vesicles isolated from those cells that are expressing the reporter with late 7 sites, and we find in both cases with B and A reporters we have the similar export of microRNA late 7 itself in extracellular vesicles, but not happening for the other microRNA that are not used for the repression, like 16 and 24. So we can say those microRNA that are used for the repression are more preferentially getting exported out, and that's why there is a lowering of cellular level of this microRNA happens after the usage. So there is a preferential export happens both in HuR and MD on cells. And we also measured the relative level of EV-associated microRNA for other contexts. For example, use a BIR1 to 2 as a microRNA to see its export happens in a inducible reporter expression system, that it happens only for the 1 to 2 reporters, but not happen for the late 7 reporters. That means this is a liver-specific microRNA exposed exogenously in the HeLa cells and only get exported when it had its target presence in presence of its target, but not in the presence of another reporter without the target site or a different microRNA target sites. So reverse is true for the late 7 also. It only get exported out in presence of late 7 reporter, but not in the presence of mid-1 reporter. So target-driven export is microRNA specific, as well as it depends on the binding sites as well. And this is the analysis of the EVs we isolated. These are the particle tracking analysis, and this is the atomic force microscopic analysis we have done to characterize those EVs that are released by the cells not expressing the reporter or expressing the reporter. There is no much change you can see in the EV size and shape when you have the reporter, but their content get different. That means their late 7 microRNA content get different when you have a reporter present in the cell. So based on that, we further experiment to find that what are the factors that can control this kind of target-driven export. So far we have the model, the target RNA when present, they somehow facilitate the export of the microRNA, but there are issues. For example, on the ER, we have shown in our previous paper that the ER or endoplasmic reticulum surface as the sites where the microRNA repression occurs, and then it's further traffic to the endosomal membrane where the Argonaut get uncoupled from the target mRNA and it is recycled, or in the other way, the Argonaut microRNA complex and the mRNA is interact with the GW-182 family of proteins, and these GW-182 family of proteins bring them to the degradation or storage compartment. We name it at GW-182 bodies or P bodies. for their further storage or degradations, depending on the type of miRNA or the protein factor they interact. So we want to check whether the GW122 proteins, by interacting with the Argonaut, can induce the depression of target mRNA for the protein synthesis, can also regulate the mRNA localization to P bodies, have a role to play in this kind of target-driven microRNA export. So we did experiment with the siRNA treatment. TNRC6B means is GW122B, also known as TNRC6B. And when you do this siRNA treatment, you can see an increased level of EV-associated microRNA occurs. So that means GW122B must act as a negative regulator of the export of microRNA via EVs when we are talking about 3 different microRNAs. All of them are going up in the GW122 negative or reduced background. Similarly, we see that the other siRNA didn't have much effect, except few of them, like SMPD2, which itself should show a decrease because it's a positive regulator of export. On the other hand, if we do this kind of northern blot analysis, you can find a similar results. And we can say that from this data that GW122B knockdown increases the EV-associated microRNA. On the other hand, we've also measured the cellular level. I didn't have data here for the quantitative data. So GW122B enhances the cellular microRNA levels, which it's also shown here. So we can see that this is important regulator of this export process that we have described so far. And when you use the target RNA, which is actually show a export, and when you have the GW122 expressed in a tagged version, in a NHA GW122B is expressed there, they prevent this kind of export that we used to see in the for the late 7 microRNA when in presence of its target. So you can see the decrease is actually rescued when we express this protein there. So from that, we can say that this is a negative regulator of export, and this is the protein. Another version of the protein is there, TNRC6C, and we use a different truncated version of that to identify the domain that are responsible, and we find that the full length is required, but and this whole part is required for this kind of interaction, which is known for the interaction of the ago protein, is responsible for this kind of lowering of EV export of late 7 microRNA when expressed in HeLa cells. So from this data, we can conclude this is a negative regulator and it's prevent the target-driven export of the used microRNAs. And we further show that the targeting of the repressed microRNA and the Argonaut protein to the P bodies is also very much affected in presence of siRNA against GW122P. So average number of AGOT and RCK bodies co-localized per cells actually got decreased when you have the GW122 siRNA expressed there. And from that way, you have the similar results observed here. So GW1-mediated microstabilization needs the P body structure, because if we use an siRNA against the P bodies and deplete the P bodies by targeting different protein factor like XRN1 or DC1, what are the constituent of the P bodies, you disrupt those P bodies, and in that case, you don't find a decrease of microRNA export that are mediated by TNRC6B is not happening. So you need that to GW122 broadly protein as well as the intact P bodies for their action of restricted microRNA export. So GW122 can restrict the cancer cell proliferation by inducing senescence by the late 7 microRNA upregulation. That is another observation we had, because this export is somewhere linked to the senescence status of the cells, because late 7 microRNA is very much important in the regulation of senescence status, attending the senescence status. So if you play with the late7 level by restricting the GW122B proteins, you can really stop the cell proliferation by doing so. So far we discussed about the GW122 proteins, how it prevents the microRNA export process, but what are the factors that are responsible for the microRNA export to occur? So in this context, I like to highlight the paper we published earlier in EMBO reports to show that the HuR protein is one of the protein that is required for the microRNA export to happen in human cells. It's interesting to find that under the normal situation, when the cells are normally grown in normal media, their microRNA is very much active and replace the target genes. But when you stop the cells, there are some genes need to get de-repressed from the microRNA mediated repressions. In that context, what happens that a protein called HuR, which is an otherwise nuclear protein, gets shuttled to the cytoplasm and did few things. One of the things they do, they bind the UTRs of the mRNA with AU sequences. That means they can bind specific mRNAs, and by binding the specific mRNA, they prevent the microRNA binding to the target mRNAs. And by the way, they replace the microRNA piece. But this is one job they do to ensure the translation of the microRNA repressed messages under the stress condition that are having HuR binding sites. On the other way, it's also uncouple the microRNA from the microargonaute proteins, and it remain bound with itself. And this microRNA bound HuR then shuttle to the endosomes, where they get ubiquitinated, and this ubiquitination is associated with the loss of microRNA interaction of HuR. And these HuR released microRNA are now become part of the endosomes and get exported out as part of the EV mediated export process. The whole mechanism was explored by Komolika in the lab, and we published a paper in EMBO report to show that the HuR is a positive regulator of microRNA export. And so far we discussed two proteins, GW-182B, which is a negative regulator of microRNA export, and HuR, which is a positive regulator of microRNA export. Now we want to see whether HuR does GW-182B affect the HuR mediated export of microRNA in the human cells. So we analyzed few data on that, and we have done experiment expressing HuR to find that HuR indeed lower the microRNA content as expected in the cells. But even if you express the GW-182B, you cannot rescue them to the normal level when there is no HuR expressions. So that means they must act in an independent way. They are not cross-talking to each other in that sense. So we can come up with that model. In the case of normal target-driven microRNA export, GW-182 act as a negative regulator of the microRNA export because it brings the microRNA, argonaute, and the repressed mRNA to P bodies and restore them there. So yeah, HuR is probably not seeing them in the normal situation. But when HuR is in action, like in stress cell, they bind the microRNA and replace the argonaute protein in the ER membrane itself, and then in the MVV's bodies, they are getting ubiquitinated, microRNA released and get exported. So these are the two pathways, major pathway and auxiliary pathway, that are operating in human cells to keep a balance of the microRNAs. In normal situation, the microRNA level gets checked by GW-182 as a probably to stop the extra export of microRNA, to prevent the loss of microRNA functions inside the cells. But in the otherwise abnormal situations, HuR take be in actions and try to lower down the cellular microRNA for different reasons. Maybe for different reasons. We have done experiment in different contexts, like in normal cancer cells and also in other kinds of cells like macrophage, and we have different observation to relate different physiological contexts of this HuR-driven microRNA export. So that paper was published in MCB last year to show that GW-182 protein restrict extracellular vesicles mediated export of microRNAs in the mammalian cancer cells. So to that context, I just like to highlight one of the work we also published that almost in the same time in Journal of Cell Science, to highlight the importance of the mitochondrial control of mTORC1 activity that are linked with the compartmentalization of a translation initiation factor EIF4E to regulate extracellular export of microRNA. So in this particular paper, we have shown that in the normally proliferating cells, when the mitochondria is very much active, it helps the compartmentalization of the mTOR, active mTOR complex to the ER, and that itself allow the EIF4E BP phosphorylations and EIF4E activity to ensure the proper translation to occur and the rapid microRNA turnover. In case of senescent cells or the cells which are deprived of some growth factor, due to mitochondrial activity lowering, there is a reduced mTOR relocalization to the ER membrane and subsequent to loss of EIF4-EBP phosphorylation, release to the translational depression and lower microRNA turnover that happen in less proliferating cells, cells with dysfunctional mitochondria, and we could find that it is without much microRNA compared to the normal cells. So that is another way of regulating the extracellular export. In that case, mitochondrial activity is coupled with the mTOR activity, which is linked with the microRNA export to occur. So far we talked about the cancer cells primarily and all these kind of senescence and other kind of thing. But there is another interesting cells that are also placed in the system for inflammatory cells, and we like to highlight now the exosomes, how they control the inflammation process and also the metabolic inflammation per se. So one of the questions we also try to address the how the metabolic inflammation that happens in the high fat exposed mice or human even in the liver itself. So you all know the liver has different kinds of cells, the parenchymal cell as well as the macrophage cells. So when we expose the animals with the cholesterol, we find that hepatocyte-specific microRNA levels in the tissue-resident and immigrant macrophage in the mouse liver and associated with kind of inflammatory response. So what we did, we fed the mice with high fat and then isolate the liver cells and fractionate them. So I sorted them in the different kind of cells, RHM and the HuR cells population. In both cases, you can identify the increase of a microRNA 122 there, which is a liver cell specific or hepatocyte specific microRNA. Supposed not to be there in these macrophage cells. But when these cells are analyzed for the precursor of the microRNA, you didn't find any increase in the precursor level of this microRNA. Only the mature form you can detect. And you also see some associated increase of inflammatory cytokine expressed there. So we want to check whether this expression of these cytokines that happen in those cells, the high fat liver, is somehow linked with the increase of the miRNA presence in these macrophages. So to mimic the situation in the cell culture model, we use the hepatic cells, HuF7 cells, and treat them with cholesterol and isolate the EVs and analyze the EVs and the hepatic cell itself to find whether you can find any correlation of microRNA in the part of the exosomes. And you can see this miR122 and other microRNA 22, 16, or almost most of the microRNA are getting packaged in the EVs that are present in the HuF7 cells and released there. And if you use that mouse primary hepatocyte derived EVs to treat the murine primary macrophage, you can see after the EV treatment, there's an increase of TNF alpha, IL-6 or IL-1 beta that are not there even without the cholesterol treated EVs or control EVs. So that suggests us there's something maybe there in the EVs that are doing the jobs, and whether it's miR122 or not, we like to test that. And we also did experiment to see that relative level of cytokines after EV treatment, and with cholesterol we have the increase of the cytokines there, and the relative level of miR122 also getting increased when you have the with cholesterol treatment. 16 level doesn't change too much, but 122 is more specifically increased. So we like to see whether this increased miR122 is associated with the increase of these cytokines. So, and we use experiment to show that if you use the anti-miR122, you can reduce this kind of cytokine production. So these are linked with the miR122 production, but the point is that how do EV get transferred through the tissue matrix to reach to the macrophage? And that's one question. We like to identify the way it can travel through the matrix of the liver. So to mimic the situation, we use a material dual chamber assay. In the bottom, we have the recipient non-infected cells, and in the top, fraction, we have the EVs isolated from the HuH7 cells, either transferred to the CY3 version of miR12, which can be visualized in the microscopic way, or CD63 GFP, which is an endosome EV-specific marker protein. And we can follow the transfer of this content in the recipient cell when the two chambers are separated by a semipermeable membrane. And we did the similar kind of analysis of the EVs. We are going to treat the cells, and we did the microscopic picture analysis for the CY3 miR12 as well as GFP per cells, and these are the microscopic data. From that data, we analyzed that the recombinant in presence of control, we have transfer, but that transfer is getting inhibited in presence of a chemical called ARP101. I'll introduce that chemical later. But this ARP101 is a known inhibitor of MMP2. And if you do the same experiment in the SI-MMP2 negative background for the donor cells, you see that there is also a reduction of the CY3MN2 transfer as well as CSD63GFP transfer, but that become increased or better in presence of recombinant MMP2 you introduce into the medium. So MMP2 is a matrix metalloprotease which is known for its role to digest the matrix protein to make a way probably for the EVs to move across the medium. So you can detect the presence of the MMP2 into the EVs primarily that are released by the hepatocytes, as well as you can see a reduction of the MMP2 in the SI-MMP2 treated cells that can account for the reduction of this transfer process. So MMP2 inhibitor 1PR also prevent this kind of transfer, and we did the experiment in another context. HuH7 cell treated EVs are now used to treat the HepG2 cells, which is another hepatic cell without the miRNA 1 to 2 expressions, and you can see the transfer of miRNA is very much there, but it gets blocked in presence of anti-miR122 molecule or ARP101 as an inhibitor of exosome. So MMP2 inhibitor restrict the functional miRNA transfer to target cells, and the recombinant MMP2 enhances the miRNA transfer process evident from this data. So we can conclude that matrix metalloprotease 2 is required for the EV-mediated miRNA transfer, and we now like to check how in the inflammatory context how it happens. So in the other situation, when you use the hepatocyte-derived EVs, we use the HuR cell to do the analysis of the transfer process. But here now, if you use a recipient macrophage cell, almost the same result you will see that miR12 will get transferred very much in presence of the recombinant MP2. But if you use the ARP101, you have a reduction of the transfer process. And if you use these EVs isolated from a HEPA16, the mouse hepatocyte cells grown to express the miR122 containing EVs, and now you incubate the EVs with the ARP101 or without the ARP101, and after the injection you sacrifice the mouse. After 5 days, you can see the level of AMP122 is very less when you have the inhibitor, but otherwise a substantial amount of 122 you can detect in the macrophage after you isolate up from this mouse liver. And the relative level of iron, interleukin 1 beta, TNF alpha, or IL-6 all are increasing when you have the miR12 treatment, but all are getting inhibited when you have the ARP101 is there. So that can suggest that MMP2 is required for the hepatocyte miR12 containing EV transfer across the extracellular matrix to the macrophage. So this is the model we have in a normal situation. When you have the MMP2 is active, the MMP2 helps the released EVs from the hepatocytes to reach this macrophage and get them activated. So when you have the liver under stress or kind of hepatic kind of metabolic stress, you have the release of miR122 via EVs, and these EV122 as part of EVs reaching the resident macrophage and activate them to probably to induce the metainflammation processes in the liver itself. And then if you have the MMP2 inhibited, it cannot transfer across the matrix to reach the hepatocytes, reach to the macrophage and activate them. In that way, no digestion of extracellular matrix happens because the OMP is inhibited, no EV targeting of the macrophage occurs, and the macrophage activation and no tissue inflammation was detected both in the mice context as well as the in vitro cell culture model. So this is a very interesting thing. So how the transferred microRNA become active in the recipient cells is another interesting question. So so far I talked about the MMP2 part. It's now published in the iScience. Now I'm going to switch to the other problem that we are addressing, that is how the transferred microRNA become active in the recipient cells. So far we discussed two aspects: how the microRNA getting exported via AVs, what are the mechanism. Few mechanisms I already discussed that are there: the HuR mediated export and GW mediated reciprocal regulation. Then I discussed about the how the EVs move across the matrix, where you need the MMP2 to restrict the movement. But the most important questions may be also there, that how the EVs carrying the microRNA deliver to the target cells and get them activated for the microRNA. How the microRNA getting part of that. So address those questions. We have few questions dissecting the pathways. So from how the microRNA and loading into the recipient cell argonaute protein. So these are the EVs with the microRNA, and they must reach the cells and the targets like endoplasmic reticulum, where the target mRNA is sitting and getting translated, and they must reach there to replace the process. What is the site of localization of this interaction of this microRNPs or the argonaute and the microRNA in the recipient cells, and the route of internalization of the microRNA in the recipient cells? So these are all questions that we want to target. And to do that, we use the HeLa cells again, and which express the CD63 GFP for the microscopic analysis of the EV transfer, and a miR122 encoding plasmid. So miR122 is not otherwise expressed in the HeLa cells. So that's why whatever EVs we are getting from HeLa cells should not have miR122 unless it's expressed exogenously there. And the same EVs now we can treat to targets another HeLa cell which doesn't express this miR122 and can follow the transfer of the miR122 through this kind of analysis. And of course, the EV you can use for the other kind of biophysical analysis as well, whether there is any change or not. And you can treat the recipient HeLa cell to visualize the uptake process and monitor the kinetics of it. And you can do the RT-PCR analysis also to identify the amount of microRNA transfer also. So we start that same experiment here, and you can find this miR122 positive EVs can transfer the actual microRNA to the recipient HeLa cells. Pre-microRNA level also change a little bit, but relative levels of microRNA remain unchanged when you treat the DNase treatment for the EV. So that suggests it's a RNA from, not the plasmid itself, getting transferred. And when we do the RNase protection assay, we confirm that the microRNA 122 is actually get trapped as part of the EVs. And then we also analyze the CT value of the microRNA and the antisense stand of the microRNA to confirm that is the mature form only get coactive to the EVs, not the other form and not the antisense stand also. So copy number also get determined. And in the recipient cell also you find that the relative level of pre-miR122 remains unchanged. Only the miR122 itself is getting detected, not the antisense stand of it, and the copy number also remained quite substantial. So mRNA microRNA getting trafficked from a single standard form from the donor HeLa cells to the recipient cells, that we can confirm from this data. Then we try to find different kind of assay to find the kinetics of the thing. Is actually at 16 hours you have substantial amount of miR122 to get transferred, so we settle with the 16-hour time point. And with the relative level of microRNA we measured, it's not only miR122 specific, but if you use an exogenously expressed another microRNA like miR146, you can see the similar results. So it's not really miR122 specific event, but other microRNA also can do the same thing. To prove that the functional microRNA transfer of microRNA are functional in the recipient cells, what we have done, we use a reported assay with a RENILA with one microRNA 122 target site. Without the EV transfer, you cannot see much changes in the activity after the treatment of the EV. But when the miR122 content EV are treated, you have a substantial repression level. and relative level of microRNA or target mRNA, that are endogenous target of miRNA 1 to 2, is also already getting reduced when after the mir 1 to 2 EV treatment. So suggests the transport microRNA 1 to 2 from the HeLa cells is become functional there, can target their endogenous gene, and the reporter repression also occurs. So to address another questions, whether the recipient Argonaut cells, Argonaut of the recipient cell actually getting loaded with the recipient microRNA or the transport microRNA, we did a pull down assay with the flag HeAgo2 expressing HeLa cells as a recipient cell, and we want to do containing 1 to 2 are co-cultured or even EV treated. We see there is an AGO-associated miR1 level increases in the recipient cells, and also the target gene level decreases and the repression level goes up. So that all are suggesting that the AGO2 association of EV-derived microRNA actually happen post transfer. But AGO is easily get loaded by this TRBB dicer complex, as I mentioned earlier. So Argonaut with the microRNA get loaded by the dicer complex. So this is a canonical pathway by which the microRNA are processed and loaded. But in this case, the microRNA is getting inside the cells as a single stranded form, not as a double stranded form. Otherwise, they are used by dicer to load the Argonaut. So the questions comes whether dicer has a role to play in the Argonaut loading of the EV-derived microRNAs. So we do find that an increased association of Argonaut happens over time. But if we use the siRNA of AGO2, we have a reduced of the microRNA uptake in the cells. But if you use this NHA tagged version of other Argonaut, you can also see they are also getting loaded too. But if using a dicer siRNA, we can see it's no much change of the microRNA loading of the recipient Argonaut that happened. Neither the total content, neither the AGO-associated microRNA content getting affected by the SI dicer, whereas the endogenous microRNA that are expressed there that need dicer for its processing are getting reduced in amount. So the internalization of the EV microRNA is AGO2 dependent but dicer independent. So it's a non-canonical way by which the microRNA derived from the EVs are getting loaded to the Argonaut of the recipient cell that doesn't need dicer in place. So as we know that the endocytosis process is very much involved for the uptake of different cargoes from outside the cells to the inside. So we want to check if the endocytic pathways are involved in the internalization process. So what we did, we used the siRNA dynamin 2, which is very much dependent for different uptake process, and you can see there is a reduction of microRNA uptake happens in the dynamin 2 level, as well as the activity of the recipient microRNA level decreases. So internalization of the microRNA uses clathrin independent and both dependent dynamin dependent pathway for the internalization. We use different inhibitor of the clathrin pathway and see that they are very much needed. And we later try to understand the mechanism by which they are getting there. So we did the ultrasound division to see the EV pellet, but we can also do some kind of fractionation analysis of the cell extract on a density gradient to separate the different organelles based on their density on a 3 to 30% iodoxonal gradient. And this OptiPrev or iodoxonal density gradient allow us to separate the different subcellular compartment of the recipient cells to analyze how they are loaded with this Argonaut or not. When you do this kind of analysis using different marker proteins like calnexin-A1 and HRS, we can separate different compartments from which are relatively enriched for the early endosome, late endosome, or the 7 to 8, 7, 8, 9 fraction is more is the ER fraction. And with time, we find that the microRNA goes up regularly, and in the ER fractions, both AGO-associated as well as the total microRNA level were 16 hours of time, but they must go through this kind of endosomal maturation before they should reach the ER. So internalized microRNA target to the endoplasmic reticulum finally, but they must go through this kind of endosomal maturation process before they enter. So we want to check whether how the different component of the endocytic pathway are contributing in this process of microRNA internalization. We have different siRNA as RAF proteins. This RAF5 is endosome, early endosome maturation requirement. RAF7 is late endosome maturation requirement, and RILP is a lysosome and late endosome interacting proteins. And with those things, we can see there is a substantial level of microRNA repression level change in the siRNA treated the RILP, but the relative level of microRNA actually goes very high when the RILP is not there. So this is very interesting. You have an increase of microRNA level, but you have no increase in the activity, rather a decrease in activity is actually happening. So this similarly thing we can find here, but Argonaut associated microRNA level goes up. That means the microRNA get internalized, they get interacted with the Argonaut protein, but they cannot probably go to the target mRNA to replace them. Why it's happening so? So we don't find any major change in the endocytic pathway component in the morphology or any other things. But what we do with the aggo-associated microRNA level we measure, we find that the both in RAF7 and RILP knockdown cell, you have a substantial increase of aggo-associated microRNA, and differential subcellular localization of microRNA on the blocking of the endocytic pathway has been evident from this analysis. So this is the picture we were ended at that time, that RAF5 inhibiting this, RAF7 is inhibiting this. But this endosome maturation, if you loaded microRNA need to go to the ER for the repression to occur, there must be someone get stuck here and could not reach in the endoplasmic reticulum for this activity, although they already get loaded with the thing. So how it get controlled, how the endoplasmic reticulum maturation in the endosome maturation process is controlling this kind of microRNA loading to the Argonaut protein. So it is known that the pH can control the two-step uncoating on influenza of virus or endosome plasmic reticulum endosome interaction. This also much depends on the maturation process is controlled. So low of pH change probably have a role to play in delivering this microRNA to the ER on the recipient cell. So we try to address the questions, and we use a drug called bacillomycin, which will block the pump, pH pump, and allow the pH not to generate inside the endosome during its maturation. And you can see the bacillomycin itself, like the RILP does, can increase the aggo-associated microRNA level, and the bacillomycin prevent the acidification of the lysosome and interferes with the aggo2 microRNA interaction. So we use another interesting assay to really propose that what could be the exact mechanism. So we use the bacillomycin treated cells EVs, which now should have a higher pH than it should, and then treat these cells, isolate these EVs and treat them with FCCP and at different pH buffer. Because if you use different pH buffer, you can change the pH with the FCCP drugs because it's subtle across the membrane after binding the hydrogen. So you can see that with that data, you can see the release of microRNA, if any, they are due to pH change in the vesicles. So we did that experiment, and we find that yes, indeed, there is a change in the pH happens. The size and the number of endosomes doesn't change. But if you use the pH 5 buffer in presence of FCCP, you can the CT value of the miRNA to lowering down. That means a lot of microRNA get released when we use the supernatant of the cell after supernatant of the EVs after the treatment of after FCCP. So this is a little bit complicated experiment, and we do the time course experiment also to suggest the release of microRNA from the endosomal cavity to the surface after the lowering of pH happens. And we did similar experiment to prove that the Argonaut present on the endosomal surface is actually getting loaded with the Argonaut protein. That is evident from this experiment here. So microRNA bind to the endosome membrane bound AGO2. That's why it can explain the process. So in nutshell, this is the process. So at pH 6.5 to pH 5 to 4, when the pH lowering is happening, the endosomes which are internalized, the EVs that are internalized are fuses with the endosomal membrane to release the microRNA. And this microRNA is now getting loaded to the Argonaut protein present on the endosomal surface that are not otherwise microRNA loaded. So these microRNA Argonaut complex form, and then they go to the endoplasmic reticulum for the repression to occurs. So if you somehow block this process of endosome maturation with bacillomycin or RILP or RAF5, you can block this kind of Argonaut microRNA complex trafficking further for the lysosomal degradation or for Argonaut loading. And you don't need dicer one to do this kind of job. It's a dicer independent process. So we can say that internalization and AGO2 association of the single stranded microRNA happens. Probable requirement of the lysosome for the formation of silencing in complex is there, and dependence on the pH for release of EV content into the ER of the recipient cell also happens. So this is the intercellular microRNA transfer, and we already discussed. I probably don't have time today to discuss other things, but just to mention that we are also working on another aspect called intercellular microRNA transfer and infection. Intercellular microRNA transfer in the infection context, and in this case, the EVs released by the leishmanias also play a role to control the host cell microRNA machineries, because these EVs that are released by the leishmania is a pathogen which interact with macrophage and remain there in the host. Also release exosome with another protein called GP63, which cut the dicer and prevent its maturation, prevent the microRNA maturation in the target cells. So that's initially we have described, and later we find that this GP63 used by the parasite is also in another context to cut HuR also, because HuR-driven export of microRNA happens in the activated macrophage, and that is required for the macrophage activation process itself. And leishmania, being a pathogen, don't like the activation of the macrophage, and it target the HuR by targeting GP63 and target the HuR to cleave it to restrict the inflammatory export of microRNA and expression of inflammatory cytokines. So that way, this is another paper we published to show that the HuR exporter microRNA exporter HuR clears the internalized pathogens by preventing the pro-inflammatory response in infected macrophage. So if you overexpress HuR, which can overcome this GP63 action, then it can rescue this kind of infection process. So the last but not least, I'd like to highlight very recent work that we have published to show that this kind of EV-mediated crosstalk is not in one-way thing, but it can have a multiple facet. For example, the macrophage which is infected with the leishmania exporting a 1 microRNA 146 from the macrophage itself, and this 146 being an anti-inflammatory microRNA go to the hepatocyte to stop the miR122 production there. This is pro-inflammatory in nature and prevent the macrophage activation in the surrounding cells for the M1 macrophage polarization thing. But on the other hand, this EV-mediated packaged 146 go to the macrophage to polarize them to the M2-like state to lower down the TNF alpha and or IL-1 beta, but IL-10 remain high, and HuR level also get lowered by the miR146. And this is also a very reciprocal way of regulation that happened in the neighboring cell, very recently published in Life Science Alliance. And this is the future. So far we have discussed about this EV-mediated packaging mechanism and so on. There must have some specificity associated with the microRNA packaging. HuR can explain part of it, but not all of this. There are other mechanisms machinery may ask to operate. So identify those things. We have already developed some kind of in vitro assay system to use some cell-free assay to use that for the microRNA packaging analysis or export analysis to do. We use the endosome enriched fraction for doing this kind of assay and standardize them. Maybe in the next time when I'll have a chance to talk, may have a chance to talk in great details about that, for not for today maybe. So before I end, I like to highlight the other wings that we are working on. For example, we have only talked about the the regulation of intra and intercellular microRNA transport primarily in this talk, but the lab is also working on the alteration of microRNA function in the pathogen-involved macrophage. So this also I partly talk cause and consequence, but I didn't have talk on these two parts. The importance of subcellular organelles in controlling the microRNA biogenesis and activity, which lab is very much also involved in this aspect. And more interestingly, we are also working on the microRNA activation and deactivation process in differentiating neuronal cell. We are working on the mechanism and the action and the consequence in the aging process and the neurodegeneration itself. So this is the acknowledgement slide where we have the collaborators who are very much involved. We have active collaboration now with Michaela Zabolan's lab and Edward Bartow's lab in Switzerland and France on the host pathogen interaction aspect, as well as on the microRNA mRNA trafficking context. And we have some collaborators in India as well, all of them from IICB at the moment. And we have those members already passed out, but they contributed substantially in the work we have done so far. Today I primarily talked about the work of few people like Kamalika's work. We also talked about the work, last one, Bottika is actually involved in this RNA microRNA uptake and its mechanism process, and Orno and others are also involved in the process of describing the work on the MMP2. And I didn't have chance to talk to the others work, but again the NCB work that I presented, initiated by Shobhik, and then Kamalika, Jogaditya are all take part into that. So these are my funding bodies, and I am very much indebted to Wellcome Trust initially and Human Frontiers, and now CEFIPRA also. But different government funding agencies like DST and DBT are very much supportive, and also CSIR for the different intramural funding we received from CSIR. So thank you all, and I will happy to answer the questions that you may have after this talk. Thank you.
Thank you, Dr. Bhattacharya, for this talk. And the session is open for questions. So anyone who wants to ask a question can just raise a virtual hand or post their question in the chat box.
Okay.
Yogita?
Hi, Dr. Bhattacharya, this is Yogita, and
Yes.
thank you so much for this wonderful work, very brilliant work. So I have a couple of questions like, so miRNAs get packaged in the extracellular vesicles as soon as they get synthesized, or they first get stored in P bodies and then they get packaged? So what is the mechanism?
No, so what we have find that the microRNA processing or biogenesis to the mature form happens also on the endoplasmic reticulum, and almost probably immediately they are getting involved in the repression process of the target genes. So and depending on the usage of the microRNA, they can be either stored to the P bodies or get exported out or even recycled. So these are the three fates it can achieve. It can go to the P bodies for storage of the mRNA and the microRNA. It can go to the packaging to the EVs, extracellular vesicles, to the endosomal pathways, or even it can go through the lysosomal degradation pathway. So which are the real player and what are the factors actually control this thing is not entirely clear. So GW-182 and HuR found to be the important player to control this kind of UV mediated export and restriction of the UV mediated export by targeting them to the P bodies. They do in opposite way. But how about the other factors that are playing a role in this kind of microRNA fate determination is not entirely clear. So that we will still need to investigate and explore.
And how the stability of miRNA affect the export process as well as the activity of the transferred miRNAs in the recipient cells? Because stability of mRNA is a crucial issue in the cell, especially in vitro cultures.
Yeah. So, but as you know, the mRNA is usually very small in size. So sometimes there's the advantage they also have to get escape from the exonuclease, because most of the bigger RNA like mRNAs are much prone to get degraded by the exonucleases and endonucleases. So, but there are other thoughts also. The people have thought there are some kind of post-transcriptional modifications also happen for those microRNAs that can get them packaged and exported out. But the other school of thought is that they are also part of some of the protein complex, like HuR maybe also somewhere wrapping the microRNA and getting them exported out without allowing them to get degraded by the exonucleases. So these could be other players that are part of the RNA microRNA interacting proteins also play a role in controlling this kind of export.
And it is the precursor or mature miRNA which is getting transferred?
So far in our hand, we only see the mature microRNA get transferred, not the precursor.
Okay. Thank you so much, Dr. Bhattacharya.
Thank you.
Chitra, you can ask a question.
Dr. Bhattacharya, here's my question is to do with this experiment that you had on exposing the mice to high-fat diet and then looking at an increase in miRNA 122 and following that increase in cytokines and everything.
Yes.
So I wanted to know, did you see if that entire thing, if the miRNA came down as well as the cytokine reactions also came down once you switched the mice back to normal diet, some kind of an interventional, some kind of an inducible scenario where this is a reversible scenario. So once you take away that high fat condition, the microRNA is the regulation is such that it also comes down and the cytokine response also abates.
Yeah. So I think it's a very nice question and very important question as well. So honestly speaking, we still have not done this kind of reversal experiment, the fat diet reversal experiment.
Right.
To comment on that.
Yeah, may I suggest that if you even try probably exercising the mice, for example.
Yeah, that's an excellent idea. I don't know how it will affect this kind of physical activities will be related to this exosomal export or it's a very nice idea, I must say, to see how it could affect EV trafficking or microRNA turnover even in those recipient cells and the turnover of the cell itself maybe.
Yes.
So it is nice thing to look at, but no, honestly, we have not done that experiment. Another important experiment could be how the trafficking happen ex liver, for example, go to the other kind of macrophage present on the other side, like in heart and other peripheral tissues.
Right.
So that we also have not done. So these are very interesting aspects to look at, but so far we can only comment the MMP2 is required for this kind of transfer to occur.
Right. I have another sort of curiosity-based question, and that is when you have this phenomenon where the miRNA is going from one cell into another cell, like in this case, it's somehow finding its way after the MMP2 activation and everything into the macrophage. I'm very curious, how is it that the macrophage the miRNA apparatus in the macrophage doesn't respond, like for example, the GW proteins in throwing these miRNAs out? Yeah,
so is also a nice question and very logical to think on that line. So the point is that I think either there is no such specificity about the macrophage itself, you can go to anywhere.
Right.
So, or if there is may have some specific receptor that allow them to get into the macrophage, but you have to remember the macrophage, the phagocytic activity, so maybe they are more vulnerable to get those.
Correct. Yes.
And they are not clear because they are also have the mechanism to get cleared. For example, they can go to the lysosome and get degraded, which probably definitely a fraction of it get so. Right. But on the other hand, as I discussed in the last half of the talk, how the EVs associated microRNA get released into the cytoplasms and become functional there. Right. In that case, you can see that the EV associate the endosomes or MVV associated argonaut are the target for those microRNA. They are already immediately get loaded to the argonaut present on the surface of these EV or MVV membranes. And that's why they are not have a chance to go through a export process. If there is a mechanism operate that allow the macrophage to immediately recycle those endosomal compartments more frequently, that may counteract the process of inflammation by this EV-derived microRNA.
Right.
This is also a possibility.
Yeah, so I was thinking maybe in connection with that, it might also be useful because I mean, I don't know about this thing. Would it be useful in that sense to look at the upstream regulation of these proteins, of GW, for example, GW182?
Yeah, GW182 is involved when they are already the microRNA action with the mRNA itself and then go to the repression process after they are going to interact.
I see. So it's at a much later stage.
Yes. But you are right that if you can have a mechanism of upregulating GW182, it may also counteract this kind of macrophage activation process that you have seen.
Right. Thank you.
Thank you.
Dr. Bhavtosh, you can ask your question directly. You had posted something in the chat box.
Hi, Shvinada. It's an excellent talk. So I have just two technical questions. So
Yeah.
my first question is that what is the microRNA load in the vesicles, and do these load vary depending on the cell or organ physiology?
Yeah, it depends on the cellular and organ physiology, because if you have some cell grown in normal situation to some kind of senescent condition, their microRNA load remain decreased. But it also vary for microRNA to microRNA. Some microRNA may have a copy number of 5 to 6. Some may have only 1 or 2 per EVs and so on. So probably there is quite kind of diversity as far as microRNA load is concerned. And probably there is some kind of diversity also in the EV content. So there is a heterogeneous kind of thing also present. So that also can account for the diversity of microRNA content in the EV.
And do you think that this microRNA level could be also changed depending on the environmental stimuli, particularly if you look the microbiome aspect or the diet aspect? Does that change the microbiome load, this microRNA load in the vesicles?
That is a very excellent idea, and we have very much in collaboration with Partho in the IICB to address some of those questions, how the microbiome may have influenced the microRNA content of the EVs released by the hepatocytes or non-hepatic cells, like epithelial lining cells, also gut epithelial lining cells. So the microbiome interaction, or even the microbiome itself, they release some EVs, the bacterial EVs, and they are also probably going to the system and affect. So that can account part of the gut-brain axis thing people are looking at.
Thank you, Shvinada.
Thank you.
There is one question in the chat box by Lang.
Yeah.
So he's asking, yeah, can you see it or should I read it?
I can see it. I can read and answer. So does viruses use import-export of EV-derived microRNAs in host cell? If so, those are agar-2 bound or free microRNA? Yeah, there are some reports that viral microRNA are getting part of the EVs derived with that. But whether they are part of the agar bound or not, that's not clear, or I would not say they are very convincingly shown, because it has been done through some kind of affinity chromatography based isolation of EVs to confirm that they are usually don't have any argonaut or dicer protein present in the EVs. There is a Cell paper published a couple of years back to show that there is no argonaut or dicer present on the EV fraction. So hardly there is a chance of the microRNA that you are finding in the EVs are bound with the argonaut proteins. So even in the case of viral infection, but if there's something abnormalcy happen in the viral cell for a specific virus, it is always possible. But in general, there should not be any agar-2. But viral derived microRNA or even viral infected cell derived microRNA pool already also changes with the viral infection process. Yes. Thank you.
Any other questions? So if there are no more questions, I'd really like to thank Dr. Bhattacharya for this wonderful talk. And we really hope that next time you're able to come here to our campus.
I'm looking forward.
Yeah, give it to us personally so there can be interaction amongst
more interaction. Yes. True.
Yeah. Thank you so much. Thanks a lot.
Thank you.